So maybe I should start to get worried because my chairs have a habit of not showing up to introduce me. So as you heard, my talk today is about AIRE. And during the first third of the talk, I'll just do a short overview of where we stand today in our understanding of the mechanism of action of AIRE. And during the second two-thirds, I'll tell you hopefully two news stories which will suggest that going forward we need to have a much broader view of what AIRE is actually doing. So as many of you know, AIRE is the gene that is mutated in patients that have a disease, a rare disease, which is called APSED, for autoimmune polyendocrineopathy, candidiasis, ectodermal dystrophy. Humans with an AIRE mutation have a multi-organ autoimmune disease that varies both in severity and organ target from individual to individual, although there are some commonalities such as autoimmune attack on the adrenals and parathyroid glands. Mice that have a mutation in AIRE also have a multi-organ autoimmune disease, and the severity of that disease and the organ choice also varies with the genetic background from strain to strain. Now, a very important clue about what AIRE does came simply from knowing where it's expressed. It's expressed primarily in the thymus, above all in a very small subset of thymic stromal cells called medullary epithelial cells, which make up less than 0.5% of the stromal network. Now this was important because, first of all, Jonathan Sprent had been saying for a number of years that, contrary to what one reads in the textbooks, these cells were actually important for clonal deletion of self-reactive thymocytes. But secondly, because of an important body of work that was developing primarily from Bruno Kaevsky's lab, that these cells have the particularity of expressing a large repertoire of RNA transcripts which encode what we call peripheral tissue antigens. So these are antigens associated with fully differentiated cells in the periphery, like insulin or proteolipid protein or antigens like that. And so the idea was that they might have something to do with negative selection. Indeed, if you do gene expression profiling by microarray, comparing expression in medullary epithelial cells of AIRE knockout versus wild-type mice, what you see down here is that there are many, many genes which are positively regulated by AIRE, and also a number that are negatively regulated by AIRE. And in red are hundreds of these peripheral tissue antigen genes that I mentioned. But please remember, at the end of near to the end of the talk, that there are also a number of gray genes that are upregulated by AIRE and are not genes that encode peripheral tissue antigens. And indeed, AIRE does control clonal deletion of self-reactive thymocytes. We and other groups, for example, Chris Goodnow's group, showed this using a simplified double transgenic system where we have a T-cell receptor transgenic crossed with a transgenic that expresses a neo-self antigen corresponding to what that T-cell receptor recognizes. So the data are that if we take OT2, which is a class 2 restricted ovalbumin specific T-cell receptor transgenic, and in the presence of AIRE, just the OT2 T-cell receptor transgenic, we get a lot of CD4 positive cells. If we then cross in RIP-M-OVA, which drives expression of ovalbumin behind the insulin promoter. Of course, you get expression in the pancreas, but if you listen to what I told you at the beginning, you should also think that you're going to get expression in these medullary epithelial cells of the thymus. And the consequence is that there's very severe depletion of these CD4 positive cells in the presence of AIRE. But if AIRE is not there, you get restoration of the population, and having the neoself antigen in the absence of AIRE is just like not having the neoself antigen at all. So then the idea is that these medullary epithelial cells produce these RNA transcripts under the direction of AIRE, and these are translated into proteins, processed and loaded onto MHC molecules, and as thymocytes are percolating through the thymus during their differentiation, if their T cell receptor interacts with this MHC peptide complex in the particular window of avidity, these cells will be clonally deleted and removed from the repertoire. Now, I should say that this is probably not the only thing that AIRE does. Different groups have provided evidence that AIRE might be involved in medullary epithelial cell differentiation, apoptosis of medullary epithelial cells, Treg selection, or peripheral tolerance induction, which actually many of them might be tied together in some way. But in any case, this mechanism of AIRE raises interesting questions about how AIRE operates on a molecular level. And that is simply that you have this one transcription factor that controls thousands of genes, which in the periphery are regulated completely differently from each other. Different tissues, different times, different levels of regulation. And so how does this come about? And so we and other people have been intrigued by this phenomenon and worked quite hard on it over the last 2 years. And I think we've come up with the basic answer of what AIRE does on the molecular level. And I'll show you that near the end, in just a few minutes. But I did want to show you 2 or 3 of the critical data which lead to that conclusion before I make it. So one set of experiments was a mid-scale screen of proteins that bind to AIRE. So this is just a co-IP mass spec analysis. And then when we did that, we found that actually there are a lot of proteins that interact with AIRE, more than 40. And interestingly, they fall into particular classes. Some of them weren't too surprising, like transcriptional regulation, because we always knew that AIRE was somehow involved in transcriptional regulation, or nuclear export, because AIRE is a nuclear protein. But others were a bit more surprising, such as chromatin binding and structure. There are a bunch of histones here, and especially pre-messenger RNA processing. Now that's one screen that we did. A second sort of mid-scale screen was an shRNA screen to determine what proteins are actually required for AIRE to exert its function. And this was done in cell culture with bookshelves of shRNA molecules targeting particular pathways. And by and large, we could confirm these same pathways: RNA processing, chromatin modification, and transcription. But what was perhaps the most striking was what we didn't find. We didn't find anything that was associated with RNA initiation, but rather we found things that were associated with RNA elongation. For example, members of the PTEF-B complex, BRD4, and things like that. And in fact, we could show a clear effect of AIRE on RNA elongation by going back to our gene expression analysis by microarray, and this time not looking at the average of all the probes across the genes, but in fact looking exon by exon using individual probes and walking all the way along the genes. So what you see here is just a bunch of AIRE-regulated genes in an AIRE wild type versus an AIRE knockout. And we have a bunch of different probes starting at the transcriptional start site and going further and further 3' into the gene. And on this axis is just expression value normalized for the average of each of that particular gene. And what you see is at the beginning of the gene, for the first 150 nucleotides or so, there is really expressed the same. But as you get into the gene, there's a distinct drop in the AIRE knockout mice, and that goes along the gene and is why we pick up such a big difference when we look at the average across the whole gene. So for those of you who follow these things, this is a classic indicator of what's called RNA transcriptional pausing. Okay, so here's what we think AIRE does, illustrated on this slide. If you studied gene regulation starting more than 3 or 4 years ago, what you were taught was that there are two kinds of genes. Those that have RNA polymerase on them and they get expressed, and those that don't have RNA polymerase on them and they're not expressed. But very recently there's been a real burst of activity showing that in fact there's a third class of genes which often makes up 50% of the genome or transcribed genome, or in some cells like ES cells as much as 80% of the transcribed genome. And so what these genes are is RNA polymerase goes on, transcribes a little bit, maybe 50 or 100 nucleotides, and just stops. And this is called polymerase pausing, and turns out to be a major point of transcriptional regulation. So that's illustrated here, where polymerase will bind to a particular promoter with its C-terminal domain sticking out. It's a very important regulatory domain. It clears the promoter, gets phosphorylated on this terminal tail, moves along a bit further, and then pauses a little bit to give it time for capping. Now, negative regulators NELF and DSIF come in and hold that pausing unless it's lifted by this complex of PTEF-B and BRD4. And these come in, phosphorylate again the tail, and remove this negative regulatory complex. And so what we think AIRE does is act at this point here to lift polymerase pausing. As I mentioned, this turns out to be a very important point of transcriptional control. It's where MYC has been found to operate. It's where much of the regulation in embryonic stem cells is taking place, and it's where a lot of regulation in LPS-induced macrophages is taking place, for example. So this mechanism actually explains a lot of the peculiarities that I mentioned about AIRE transcriptional control, how it can control thousands of genes at one time, because thousands of genes in any cell have pause polymerase on them. It explains how you have an enrichment for these peripheral tissue antigen genes, because genes associated with terminal differentiation are preferentially have pause polymerase on them, and then also can explain the large number of partners in different regulatory classes that AIRE interacts with, because this C-terminal domain here acts as a scaffold for many molecular processes, including splicing, elongation, and actually nuclear transport. But just to be clear, polymerase is already loaded on AIRE-dependent genes, but it's paused, and AIRE acts to lift this pausing. Okay, so that's my general overview, telling you basically what we think about the cellular and molecular mechanism of AIRE. Now, the rest of the time, I'm going to talk about new data which came up when we started studying an observation that we made a couple of years ago, which we found very intriguing but didn't understand at all. And that observation comes from a mouse line that we made, which has the AIRE knockout on the NOD background, which makes a very strong autoimmune disease. And we crossed into that tetracycline regulatable AIRE allele so that we can turn AIRE on and turn it off whenever we want to. Okay, so this is the basic background. So in blue is when AIRE is turned on. So in this transgenic AIRE knockout mouse that we have, when AIRE is on, animals survive normally. They have a small amount of organ infiltration because remember this is an NOD background, so there's a little bit of autoimmunity occasionally. Now in the knockout situation where we don't have AIRE expressed, the animals die or we have to sacrifice them because of weight loss universally, and there's strong organ infiltration. Now, if we have AIRE on and progressively later and later turn it off, at the beginning things look just like in the knockout, but we come to a point at about somewhere between 2 and 3 weeks where if we turn AIRE off, if we turn AIRE off, it seems not to make any difference at all. So in fact, this mouse here where we've had AIRE on and then turned it off at about 2 or 3 weeks looks just like the AIRE wild type. They survive and they don't have much infiltration. Now conversely, if we have AIRE off and then turn it on during this 2 to 3 week time point, it looks just like an AIRE knockout. So the animals essentially all die or have to be sacrificed, and they have a lot of organ infiltration. So what we realize is that there is a very important window, what we call perinatal window, where it's necessary and sufficient to have AIRE expression in order to avoid autoimmunity. So what could be the explanation for this? One possibility is that AIRE induces a different repertoire of transcripts in neonates. Another is that AIRE is particularly effective at negative selection in neonates. And a third is that AIRE neonates or perinates select a particular repertoire of regulatory T cells which would be absent in the AIRE knockout mice. So we believe the first two are incorrect, and I'll just show you some quick data on that. So if we do microarray analysis comparing the adult AIRE knockout versus wild type versus the perinatal AIRE knockout versus wild type, this is always on isolated mechs. If AIRE is creating a particular repertoire in the perinates, we would expect to find genes in this region here. However, that's not the case, and we see things essentially along the diagonal. We also know that negative selection isn't particularly good in perinatal mice. This is the same type of experiment that I showed you a few minutes ago with our double transgenic OT2 RIP-M ova system, and I showed you that you got in the adult heavy deletion of the OT2 T cell receptor transgenic T cells. Now in the perinate, that's not much better. In fact, if anything, it's a little bit worse. So then we concentrated on this third possibility here, which was that there might be some special population of regulatory T cells selected in perinatal mice. So the first thing we did was to take our AIRE knockout mice on the NOD background, which get a very severe autoimmune disease, and look carefully at selection of regulatory T cells. Now, we had shown several years ago that in adults, we didn't see any difference in Treg populations in wild type versus AIRE knockout, so we weren't very warm to this idea. But this time we concentrated on this perinatal period here when Tregs are just emerging. And we see quite clearly in both the thymus and the peripheral lymphoid organs that the selection of Tregs is compromised in the AIRE knockout mice. And the next experiment we did was to try to specifically delete or ablate those Tregs which are made during this perinatal period and see what happens. And to do that, we used a mouse that's on the NOD background and is expressing diphtheria toxin receptor under the FOXP3 promoter enhancer elements, which allows us to deplete Tregs. And what we find, so in these experiments, we're always comparing litter mates, which are DTR negative, with litter mates that are DTR positive and get ablated. And so if we deplete Tregs in a young adult over this 10-day period from 35 to 45 days, we don't see much of an effect on survival, and we see only a small amount of organ infiltration. However, if we deplete them during this perinatal time from 0 to 10 days, we end up having to sacrifice all of the animals by 24 days because they're so sick, and we see a large amount of organ infiltration. So basically, we've been able to largely phenocopy the AIRE knockout mutation by depleting Tregs between 0 and 10 days of age. Now, we were a little bit worried because it's kind of tricky to give these injections to neonatal mice, and we were a bit worried that we were perturbing them. So we did a complementation experiment where we added back regulatory T cells from a wild-type mouse, and we could pretty much complement the effect of depleting the Tregs during this early period, unless we took the Tregs from a mouse an adult mouse where we had depleted the Tregs perinatally, and then we were not able to complement. And then just the last set of studies on this topic use a mouse which we were fortunate to get from Sasha Rudensky, where he made a reporter mouse, a lineage reporter mouse, where all of the regulatory T cells express GFP, and we can specifically label regulatory T cells during particular time windows using tamoxifen. So, and we cross this onto a NOD background. So, first of all, what we did was to show that Tregs that were labeled in an adult mouse were stable over time. This is 1 day after the labeling, a week later, and 8 weeks later. Same thing if you label them perinatally. So this is a mouse where the cells were labeled perinatally, and we could pick up these cells in pretty much the same numbers 8 weeks later. The other point was that, yes, indeed, this population of perinatally Tregs was absent in the AIRE knockout mice. So we wanted to use this system then to see what's special phenotypically about these perinatally generated Tregs. So the kind of experiment that we did was to tag Tregs in a perinatal mouse versus in a young adult mouse, and then carry the mice, both mice out to the same age, months later, and then study the perinatally tagged versus the adult tagged cells. So the first thing that we could show was that these perinatally tagged Tregs have a distinct function. And in this experiment and all the experiments that I'm going to show you on this topic, we're doing a four-way comparison. So the GFP, YFP are the ones that were punctually tagged. So we have ones that were tagged perinatally. And then we have, from the same mouse, just bulk Tregs which are GFP positive and YFP negative. So it's a negative control. And then two other parallel negative controls are the adult tagged Tregs and the bulk population. And so we transfer these Tregs that we isolate using GFP or YFP to isolate them. We transfer these neonatally into an AIRE knockout mouse. And what we see is that only in this case here of the perinatally tagged Tregs do we protect from the disease and protect from multi-organ autoimmune disease. All of these three negative control populations were not able to protect from the disease. Secondly, we found that the perinatally tagged Tregs have a distinct transcriptome. So here's a fold change plot where we're comparing perinatally tagged, from the same mouse, perinatally tagged Tregs and bulk Tregs versus, in another mouse, adult tagged, oh sorry, these are perinatally versus adult tagged, and these are just the bulk populations from the young and the adult mouse. And what you can see very clearly is that there are a large number of genes which are different from the perinatally tagged Tregs versus the adult, both upregulated and downregulated. And so if we start studying something about this perinatal repertoire, and for the next couple of slides I'm showing you volcano plots where if the set of genes is skewed in the perinatal transcriptome, it will fall to your right, and if it's skewed in the adult transcriptome, it'll fall to your left. And what I've put in red here is the standard Treg signature, and you can see that the standard Treg up signature is particularly enriched in the perinatal mice. And also an activation signature, Treg activation signature, is also highly skewed in the perinatal mice. And indeed, if you purify the perinatally tagged and the adult tagged Tregs, remember these are all from adult mice, they were just tagged perinatally or tagged as an adult, you see that the ones from the perinatal mice are better at regulating T effector cells. So we think the perinatal Tregs are particularly active. Now, another interesting observation, we think, came from actually looking what these genes are that are upregulated or downregulated in the perinatal Tregs, perinatally generated Tregs. And we saw some things which caught our attention, such as PPAR gamma or GPR15. And they caught our attention because some labs, particularly us, have been very interested in not the Tregs that are circulating through lymphoid organs, lymph nodes, spleen, which 95% of people study, but actually Tregs that you can find in tissues. So in visceral adipose tissue, there's a specific population of Tregs with a specific transcriptome that controls insulin sensitivity. In muscles, particular transcriptome that is actually able to control muscle satellite cells. There's a particular population that we know of in the intestine. There's another population in the pancreas. And some of the particularities of these different populations were PPAR gamma and GPR15. PPAR gamma for the adipose tissue Tregs and GPR15 for the intestinal Tregs. So we did this kind of volcano plot that I showed you before, and we see that the fat Treg signature, the pancreas Treg signature, the muscle and the colon Treg signature are all heavily skewed in the perinatally generated Tregs. So we're actually wondering whether these perinatal Tregs are what seed the development of these, what we call tissue Tregs, which are important for tissue homeostasis. Now, another particularity of these tissue Tregs that I told you about is that they have a very clonally restricted repertoire. So here we've done single-cell TCR sequencing, And if you pull single cells out of the lymph node or spleen, you can do that 500, 1,000 times, and you always get the same sequence because it's just a reflection of TCR diversity. However, if you pull Tregs out of tissues, for example, the adipose tissue or muscles, you find very impressive clonal expansions of Tregs. Now, these are repertoires that are not restricted in any way. They're not single TCR transgenics. They're just wild-type mice. Pull out the Tregs and do a bunch of single-cell sequencing, and you find these highly restricted repertoires. So when we then looked at the lymph nodes, at the adult tagged Tregs in the lymph nodes and spleen, they look just like standard lymph nodes, where over and over again you get different sequences. But if we looked at the perinatally tagged Tregs, we start to see clonal expansions. So again, we're wondering if these perinatally tagged Tregs what are the source of these tissue-restricted Tregs. And another thing that makes us think that, which is consistent with that, is, we know that in adipose tissue, this particular fat Treg that we call it comes up with age, and it really starts to seed between in this period here. So we actually did a bunch of thymectomies looking at later and later times, and if we do a thymectomy at 13 weeks, it makes no difference to this accumulation. We do it at 6, 4, and even 3 weeks, we've seen no difference in the accumulation, which again suggests that maybe these special populations of Tregs are seeded very early. Okay, so just to summarize this part, we think that not all, but a substantial part of AIRE's impact is being an influence on the selection or survival of a population of Tregs that is generated perinatally. And this population appears to be specialized in controlling autoimmune manifestations because it was only this population that we could put into AIRE knockout neonates and protect from the disease. We couldn't put in adult Tregs, at least at the numbers that we were using, which was 100,000 per mouse. And then we also wonder if this very special population of Tregs that's made perinatally has something to do with these Tregs that one finds in tissues and are important for tissue homeostasis. And maybe some of you have been following the story on macrophages, where there's particular populations of tissue-resident macrophages that are also generated very early on from fetal liver precursors. So we're wondering if we don't have a parallel story here. And then I just finish up by a second story, which turns out we think may be rather related, but we're still trying to figure out the links. So when we were thinking about what could be different about perinatal mice, another thing that came to mind was that populations of gamma delta T cells are undergoing a lot of changes during this period, and they also, the ones here, are made from fetal liver precursors. So at first we weren't too hot about the idea that they might have something to do with AIRE, because there was a paper that was published saying that there's no difference in the gamma delta T cell populations of AIRE wild type and knockout mice. And actually, when we looked at that ourselves, our results were the same. This is an adult mouse, wild type and AIRE knockout, and we see total gamma delta cells, they look pretty much the same. However, there's been a lot of interest recently in gamma delta cells that are expressing IL-17 and have some implications in autoimmunity. And what we find is that there is a clear increase in these IL-17 producing gamma delta cells in the AIRE knockout mice. That's true in the thymus, as I show here, and it's also true in the periphery. In spleen, you see an increase, lymph node, lamina propria, but actually perhaps most strikingly in the skin dermis. Now this is not the epidermis where we have these V gamma 5 positive cells, but just below the skin dermis. These differences between AIRE wild type and AIRE knockout are most striking around birth. And this known data that there are these waves of gamma delta cell production made us wonder if it's not in particular V gamma 6 expressing gamma delta cells which might be enriched in AIRE knockout mice. So if we look at the mice and use antibodies against the particular V gamma regions, we do see a slight increase in V gamma 4 positive cells, but it's clear that the majority of the increase are in these V gamma 6 positive cells that are generated neonatally. So how could AIRE have an effect on gamma delta cells? We entertained a number of different possibilities. One is that AIRE might be inducing selecting ligands. So the data coming from a few labs, including the Heyday lab for SCIT1 and the Chen lab, is that during the differentiation of gamma delta cells in the thymus, production of IL-17 is already set, and it's sort of a default pathway if they don't see their ligand. But if they do see their ligand, they become interferon gamma producing. So it would kind of make sense that if in the AIRE knockout mice we have less molecules that could be associated, that could be used for ligands. A second possibility is that AIRE might induce or repress cytokines or other factors needed for these T gamma delta 17 cells for their generation or maintenance, or for the IL-17 production of these cells, or that it just does something else. So we looked at the gene profiling data that we had on MEX from wild type and knockout mice at all of the different gamma delta cell ligands that have been described in the literature. And in fact, many of them are AIRE independent, but there's some that are AIRE dependent. Unfortunately, none of these ligands are recognized for V gamma 6 positive cells, so we don't really know how to interpret these data, but it leaves the possibility still that AIRE might be controlling the V gamma 6 ligand. So, however, we could get interesting data on the role of cytokines. What we found was that in AIRE knockout mice, there are differences in the gamma C family of cytokines. In particular, IL-7 drew our attention because it's quite heavily upregulated in the neonatal cells, medullary epithelial cells of the knockout mice, as you can see here. And that drew our attention because of this paper from the Heyday lab that said that actually IL-7 is very important in the human case for production of IL-17 producing gamma delta cells. So in fact, these V gamma 6 positive cells have high levels of the IL-7 receptor, and if we come in to the AIRE wild type mice, give them IL-7 IP, wait 3 days, we find that we increase very clearly the fraction and number of IL-17 producing gamma delta cells. Inversely, if we come in with anti-IL-7 receptor into the AIRE knockout mouse, we find that we lower the expression vis-a-vis when we put in IL-7 receptor, anti-IL-7 receptor versus not. And then lastly, we did this experiment which I couldn't believe hadn't been done until then, and that is we just simply crossed the gamma delta knockout with the AIRE knockout, and what we found was a very clear improvement of the AIRE knockout phenotype when we removed gamma delta cells for some organs, but not other organs. So it seems that these gamma delta cells are important for autoimmunity in the eye. They're also important for the lung and some other organs, but certainly not all organs. And there's clear effects on the histology. And I should say that actually we can look either by flow cytometry or histology for these T gamma delta 17 cells in the uveo retinal region of the eye, and we find them there before any evidence of the disease, and they're greatly increased in the AIRE knockout mice. So to summarize this part, it looks like AIRE also regulates the generation of IL-17 producing gamma delta cells, in particular through its upregulation of IL-7, and that these gamma delta cells are important for the AIRE-less disease. Just one last general comment trying to bring everything together. As I mentioned, I think going forward, we need to think more broadly about how AIRE works. An interesting point, I think, is that we have this one gene that's mutated and affects a number of immunological processes, and that makes sense because of what this transcription factor actually does. It affects polymerase pausing, which affects whole batteries and whole transcriptional programs. And then lastly, I want to emphasize the importance of this perinatal immune system, which seems to have very special properties and is likely generated by fetal precursors rather than adult precursors. And can I just show my acknowledgement slide? Oh, okay, so I just want to acknowledge the people who did this. The neonatal stuff was done by Si-Young. Nori did the gamma delta studies. Maria did the TET regulatable AIRE, and these other people made various contributions. And then lastly, my colleagues who have given us a number of different mouse strains and other reagents. Thank you.